Good morning, everyone. Thanks for joining our webinar session. Let me introduce myself first. I'm Anna Darrocha, European Life Science Commercialization Manager at Aventor, and I am very pleased to welcome you to today's webinar, Colony Picking and Automation, How to Implement Cell Line Development Microbiology Workflows, given by our colleagues from Molecular Devices. Today's speakers will be Jeanette Greystone, European Sales Manager, and Carola Mancini, European Film Application Scientist at Molecular Devices. But before I hand over to our speakers, I would like to give you some information about the progress of this webinar. It will be recorded, and you will receive the replay link by email following this session. There will be a Q&A session following the presentation, but it will not be a live one. So you should submit your question within the question box you see at the right-hand side of your screen. You can write them down at any time, even during the presentation, and they will be read and answered during the Q&A session. One last point, there will be a survey at the end of the session. May I ask you to fill it in? It is very important to us to have your feedback in order to improve the quality of our webinars. And finally, if you have additional comments, requirements, you can as well contact us at webinar@aventorsciences.com. This said, I hand over to you, Jeanette.
Thank you very much, and thanks for joining our webinar this morning. So let me just look at the schedule of the presentation. We're going to be talking a little bit about the basics and history of colony picking and common applications where this technique is used. We'll also discuss how this process can be automated, key considerations when deciding how to automate your process, and what kind of solutions are available to you. Finally, we'll look at a case study and a video from the Edinburgh Genome Foundry and how to scale up with lab automation. So let's get straight into it now. We're going to split this into two different sections. We're going to look at the microbiology colony picking and workflows, and then we're going to look at more cell line development based workflows. And my colleague Carola will take over that section of the webinar. So let's start with the microbiology side, a brief history. This is a subject I actually find really fascinating. And you probably, if you studied microbiology at university or at school, you'll have had some of this background before. But it essentially began the whole study of microbiology with the development of the microscope, and this was actually developed by Antony von Leeuwenhoek. I hope I pronounced that correctly. He was a cloth trader from Delft in the Netherlands, and although many others had seen microorganisms before him, he was really the first one to give a detailed description of protists and bacteria living in a wide range of environments. And then following this, in the late 1800s, the actual inception of microbiology as a direct science started around the 1850s, where you'll all know a very famous Louis Pasteur was able to demonstrate that microorganisms are responsible for the fermentation of fluids. And then followed also by the very famous German Robert Koch, who was able to culture the anthrax bacteria. So these two researchers really were the forefathers of the whole theory of germs and disease. And yes, you'll have definitely heard of these very famous scientists. So moving on to the next slide, as I mentioned, we're going to split into two different sections. And talk about colony picking in the two branches, basically mammalian and microbial. It's a very powerful tool within the workflow of mammalian cell line development and also the study of microbiology. So in things like clone screening, monoclonality verification, cell sorting, knock in and out, doing things like hybridoma cultures for monoclonal antibody production will be covered in the second part of this webinar on colony picking mammalian cells. And for now, I will focus on the microbial colony picking. We'll have a little, those in that branch, you've obviously got things like library management and screening, enzyme evolution, antibody discovery, strain engineering. So these are two very distinct branches, both where colony picking is required. So moving to the next slide, let's have a little bit of a look at where microbiology's applications occur. So it is basically the study of microorganisms such as virus, fungi, algae, and parasites. And one of the very sort of main areas that's very useful to mankind and society is biotechnology, where microorganisms are used as living factories, in particular to manufacture things such as pharmaceuticals that otherwise could not be produced. And the branch of that, synthetic biology and synthetic biology engineering, can have very different applications in medical science and in the pharmaceutical industry, but also in things like agriculture and the food industry. For example, bacteria can be re-engineered to increase plant resistance to insects or frost, and also the creation of non-dairy vegan milks and cheeses, and things like synthetic meats and biofuels are all increasingly used in society today. So there are a huge number of processes that encompass synthetic biology, and that's why modern microbiology and essentially colony picking is so important. So let's have a look at what colony screening actually is. In microbiology, and you'll all remember this if you've done microbiology at university, a colony is a group of bacteria, fungi, and other microorganisms which have been grown on a solid medium made of agar usually, and this medium can have extra nutrients and things added to it that the microorganisms need to grow. And what we need to do is usually to pick from the colony morphology a pure colony, and then we know that we can grow that on and study it. And also what you might do manually is to pick a pure colony and then regrow that in agar, or perhaps you might take like a toothpick and actually streak that plate to allow the microorganisms to grow and therefore produce discrete colonies. Doing all these processes manually by hand is an extremely subjective process. It's prone to human error. And also manual picking is very slow, it's laborious and time consuming. So it's a really interesting part of the process that we can automate and save time, give us the option to do other things whilst making it less error prone and less subjective. So if you're going to start automating colony picking, you need to kind of optimize the process. And today we'll go through what to consider to do this. So what kind of colony picker to choose, what you need to have within your colony picking instrument, some relevant factors, things like throughput and scalability, how you can track your samples, to the criteria you might use to select the clones of interest, also the microorganism compatibility, as you may work with different strains, the type of applications you want to do, and also even things like labware and the requirement of labware in your future requirements. Maybe you're going to automate the processes even further. So these are things we're considering, and we're going to go through now each one or some of these in a little bit more detail. So first of all, let's look at increased throughput and automation. Automated colony pickers offer increased throughput and water wait time compared to doing it manually. So obviously the number of colonies you can pick per hour by hand is low and the whole process is very manual. If you then add for example, a colony picker, you can probably increase this throughput. A small colony picker benchtop could maybe pick automated now about 1,000 colonies per hour. It's a small, compact, low-cost colony picker which you could have. But to achieve even higher throughput, you may want a larger colony picker that's faster, maybe up to around 3,000 colonies per hour can be picked. So now your walkaway time has increased dramatically. So it's low, not much walkaway time at all. You have to stand there and pick manually, medium or high that you need to think about. If you want to do a high throughput process, you may even consider adding additional automation. So your colony picker will then hand off the plates to an incubator or to a liquid handler and that sort of thing. So we'll look at that in a little bit more detail later. So let's go on to sample tracking and the audit trail. It's increasingly important to have CFR certification or other compliance requirements. The audit trail helps to establish the credibility of your data. and also the credibility of qualitative studies, and to serve to convince the scientific community. So if you're publishing a paper, then obviously you need to prove the rigor of your data. One of the steps used to establish the credibility and conformity of a qualitative study is the presence of one of these audit trails. So you need to be able to track a clone from kind of starting picking down through the process, where it came from to where it goes in the destination plate. So this is an important consideration, and this will be part of the software of your colony picker. Let's also look at microorganisms and compatibility. So is your colony picker compatible with the organism you're working with? For example, bacteria such as E. coli are pretty easy to pick. You can pick them with a toothpick if you're doing it manually, but other microorganisms might be difficult to transfer, and you want to get a good transfer of material. So you want to have a picking tool that enables a good transfer of material. A yeast might need a special type of pin. There's a pin there which is called a honey dipper pin, which will give you a higher transfer of volume due to its shape. And then, for example, if you're picking fungi or working with fungi, which are kind of fluffy and harder to transfer, you may benefit from a pin which is kind of shaped a bit like a Christmas tree. And this will really be able to pull up some of the sample to the larger surface area, and again, give you a good transfer of material. So this is also important when you're considering your compatibility of how you're actually going to pick the colonies. Let's then look at the clone selection criteria. When you're picking the clones of interest, it's extremely important to decide how you're going to pick them, what criteria of selection you're going to include. So this might be, for example, based on morphology. It could be based on size, shape, proximity to each other, proximity to the edge. It could be that you're also maybe using a fluorescence marker. So these are the sort of things that you will decide to clone select your picking of your clones via these criteria. And you can see in this example, there's some orange colonies which are too close together, so they've been rejected. And here the yellow dots, or the yellow ones, meet your criteria, and they will be picked and go on to pick. So this is all going to be refined depending on, as I said, colony size, shape, diameter, proximity to another colony, and also contrast to the background to give the camera the ability to allow for objective and easy colony selection. Once you've actually picked your colony, it may be interesting to decide on other applications what your applications are actually going to be. To achieve high throughput, an application-driven choice can be considered when you switch from manual colony picking to automation. Things like currently things such as strain engineering, high throughput cloning, and protein expression. This could be a CRISPR-Cas9 based approach. And also then you may, those particular techniques rely on the possibility of selecting colonies of interest also based on a fluorescence screen, which gives you an easier identification of these high value targets as well. So let's take a quick look then at fluorescence-based screening applications. So conventional methods use morphological selection, but then a fluorescence application in comparison to a conventional method gives you kind of fluorescence selection which saves on screening time, and this also focuses on downstream assays and screening of your high value targets. And you may have the requirement of compatibility with a broad range of fluorescent cloning vectors. In this particular example, there's colony screening being done here based on red-green fluorescence, and this is usually, for example, in required for CRISPR-Cas9 library generation. And have you also considered your next research steps? Future requirements. You may want to do some sort of liquid handling. The colony picker may be able to do this. It's actually to plate and streak, so that would be to deposit liquid inoculum onto the surface of the agar and then streak it onto the surface. Could then be transferred back into the incubator for the colonies to grow, and then back on the colony picker so those colonies can be picked. You may be looking at library management, so you may also want your colony picker to be able to do plate replication, so compression and expansion, or you may want it to be able to consolidate the best clones into one plate, so that could be like cherry picking or rearraying. And as I mentioned before, you may also want to do fluorescent screening. So let's just have a couple of minutes to look at some of these applications. So as I just mentioned, plating and screening, I'll just play this video. So this is an example of a colony picker that's actually, hold on one sec to play. It doesn't want to play. Okay, well, if it was playing, you'd be able to see that the tip was depositing a liquid inoculum on the surface of this bioassay tray, which has been divided into, this is a 48 region Q tray, and it will then go into the incubator and grow, and then when you take it out, you'll be able to see the different patterns. So here you can see on the 48 region Q tray on a bioassay plate, and here you've got colonies growing on an 8 region Omni tray, and on a 4 region Omni tray you can see in different patterns. So it may also be important to look at eventual requirements of automation, and I don't just mean automation of the colony picking itself, but I mean what else is in your workflow. So for example, this is a strain engineering platform that's been automated. I think this video is going to play, so yes, it's going to cycle around, and you'll be able to see that there is a lot of different instruments as well as the colony picker on this workflow. So here we need an API automation or application programming interface with a modular deck system compatible with robot integration. So the robot can feed plates back and forth between automated incubator or the colony picker, and you would usually have a special automation team that can basically tailor this kind of design of a custom instrumentation. So you've got your colony picker, you've got liquid handling there, and you've got plate hotels, peelers and sealers. So all of these things can be added to your workflow in order to give you a full platform. And this one is just an example of strain engineering. What it also could be something like a plasmid production workflow, and things like that. So it's possible to do not just the colony picker, but from end to end, the whole workflow. So let's just look at 2 examples of kind of very popular workflows. So this is a molecular cloning workflow. You all know that this is a technique to study biological traits through in vitro gene amplification. And in this workflow here, a target DNA sequence is first ligated into a prepared vector, such as a plasmid. And assembled plasmids are then used for transformation of microbes to deliver the foreign DNA into the host. Then the transformed cells are plated and spread onto agar plates using this plating and streaking functionality that we talked about, and then into the incubator. As the bacteria replicate, the growth of multiple colonies with the desired DNA sequences then occurs. And they could then be manually picked, or they could then be picked again by a colony picker, because picking them manually would be very laborious for all the reasons we talked about. And as you're probably working with hundreds of plates and kits, automating the process can dramatically reduce this kind of labor time while increasing the throughput and efficiency. And then these selected colonies can be easily inoculated into a liquid media for downstream hit identification and library management. And the second one I want to look at is a very classic phage display workflow. Again, you'll all know phage display is a method to study the interaction of a protein or a peptide with a target protein. So this tool enables the discovery of high affinity binders using bacteriophages to present a target protein on the external part of a viral coat. And to increase throughput of a phage display screening, automated colony picking can be really useful here because it can be used instead of manual methods. And this selection process then requires further validation using immunoassays such as ELISA. So you could also add a plate reader here or a high content imager onto the phage display workflow. So again, not just the colony picker, but to consider the whole workflow and what other pieces of automation you might require. So I'm just going to look at one case study now. This is the Edinburgh Genome Foundry in Edinburgh, and it's a research facility specialized in the assembly of large DNA fragments. They actually build genetic constructs for academic and industrial customers using the QPix. So the QPix gives them high throughput, accurate sample and data tracking, and they have a fully automated workflow. I'm going to show you a video in just a second. Their platform includes 3 robot arms, 2 liquid handlers, various incubators, PCR machines, an acoustic dispenser, and of course centrifuges, plate sealers, diliders, cappers, decappers, all that good stuff. So I think we're going to try and play the video now. So we're just going to briefly change presenters. Thank you, Carola. Just bear with us for 1 second. Hopefully you can see the video.
The Edinburgh Genome Foundry is a research facility at the University of Edinburgh, and we specialise in the assembly of DNA constructs. Our customers come from academia through to industry and work on a range of projects from basic biology through to engineering whole cells and organisms. We have been able to fully automate the process of DNA assembly because we have a well-integrated robotics platform. We have 3 robotic arms like this one that move consumables between pieces of equipment like liquid handlers, thermal cyclers, incubators, and other pieces of equipment that we need to assemble DNA. The Cupix 420 colony picker is an important part of our workflow, and we worked closely with molecular devices to integrate this with the rest of our robotics platform. In the next part, we're going to show you how we use the Cupix for our DNA assembly workflow. Introducing assembled DNA into bacteria or yeast is an essential part of the DNA assembly process. Our Cupix has no back panel, which allows the robotic arms to move consumables in and out of the machine. This is one of the customizations that we need for the automated plating process. Liquid E. coli suspensions are grown on solid agar plates so that single colonies containing the assembled DNA can be grown overnight. We need to use SBS standard Omni trays for the plating instead of Petri dishes because these are not compatible with the rest of the equipment on the platform. The Cupix is currently dispensing up to 8 different liquid suspensions of E. coli onto the surface of the agar. Next, the 8 pins of the plating head drop down and spread the cells evenly on the surface of the agar. The plate is then removed by the robotic arm, relidded and transferred to an incubator before loading the next Omni tray into the Cupix so that the process can start again. In our current workflow, we can plate up to 96 different samples in less than an hour without needing to move consumables in and out of the Cupix. This saves us a lot of time in the lab and has improved the throughput, reproducibility and precision of the plating process.
Okay. Carola, you can take over now and talk about mammalian colony picking.
Okay, thank you, Janet. So in the second part of this webinar, we'll focus on the mammalian colony picking, so the second branch that we have discussed on the clonal screening. Next slide, please.
Sorry,
as a brief...
Can you see the screen, everybody? I'm sorry.
Yes, we can see the screen. You just need to move to the presenter mode.
Yeah, when I go to presenter mode, it just goes black, so I couldn't see anymore. Sorry, everybody. I'll try again. Not sure why. Do you want to share your screen, Carola? I mean, I can share it like this, but...
Yes, let me share my screen.
Okay, it's just going black when I share, so okay. There we go.
Am I sharing?
Yes, you are. Thank you.
Okay. Okay, as a brief history of mammalian cell culture, we can say that the founding father of physiology was Claude Bernard, proposing that cells from living organism can be maintained in a living system after the death of that organism. That led to the establishment of, historically later on, of ATCC as a regulatory cell culture institution. And also later on with other further development of cell line in use, such as Gay establishing the first human cell line, the well-known HeLa cells, but also the work of in 1975 of Kohler and Milstein to develop the first hybridoma cell lines from the fusion of myeloma cells with B cells. That led historically also to more recent rediscoveries in 2006, when Yamanaka and colleagues obtained the first induced pluripotent stem cells, also known as iPSCs. Therefore, so mammalian colony picking will be the second part of, we will be discussing the second part of this webinar, different topics. So of course, when you when you do your cell line transfection, for instance, you may wanna be able to have a high throughput clonal screening of your cell line in use, which may be different cell types. So widely used CHO cells or HEC293, but also HeLa cells or other type of stem cells. Therefore, monoclonality verification represents an important aspect to consider when choosing automation for your mammalian colony picking, but also other complementary technologies such as single cell dispensing or print or bioprinting or single cell sorting in order to achieve a monoclonal cell line, which can be edited for instance, to achieve a cell line knock-in or knockout. And all these aspects may be also used for to screen with to achieve a high throughput screening of hybridoma cultures, also for instance to produce monoclonal antibody for gene therapy, for instance. So in the next, so what are currently bottlenecks in mammalian colony picking? So there are different challenges and solutions. Above all, time represents a critical factor because when in the needs to screen thousands of clones during the mammalian cell line development, we need to accelerate selection of high-value clones and to be able to find the rare high secreted clonal cell lines. In this case, as you can see in the graph, there is a comparison in times. So in particular, weeks to screen 12,000 clones, comparing limiting dilutions, so manual methods versus other technologies such as automated colony picking, which can help you to rapidly shorten, so to shorten your CLD, your cell line development timeline, and rapidly ensure monoclonality of the cell line that you are developing and that you are submitting for regulatory approval. And of course, another aspect important to consider for automation is the possibility of objectively assess cell confluency, cell viability, and other parameters that we will discuss later on. So therefore, what's the what's the difference between a manual process within cell line development versus an automated process? As you can see here, we have a limiting dilution workflow example where we commonly have to seed our cells and image them on day 0, and afterwards let them grow for a number of days to observe and quantify the colony outgrowth. So we want to be able to then doing some colony assessment, for instance, based on colony productivity, because we want to be able to select the highest producers, and then doing some clone quality control, so eliminate clones with poor growth, normalize productivity, and do some QC on quality assessment, for instance, and having a proof of monoclonality. Therefore, afterwards, we can go for the second round of screening, which require other 14 days, and that's why timelines are longer in comparison versus automated colony picking. In order to, so according to FDA regulatory agency, in order to hit the acceptable probability of monoclonality, with manual methods, 2 rounds of limiting dilutions are required. However, there are automated technologies that can ensure a single round of clonal screening, thanks to the recent technology of colony picking with CSAC imaging on day 0. So imaging on multiple focal plane of your plate occurring on day 0, where the planar imaging on multiple focal plane can allow you to identify a single cell and confidently select the colonies which are growing from these single cells after 14 days, and then selected clones based on the fluorescence intensity, for instance, and screen them and pick the high value clones based on what could be morphological or fluorescence intensity parameter. And this with this automated technology, you can have basically monoclonality verification integrated in one single round of screening. So what are therefore, we're going to go through a checklist of different aspects and parameters to consider for your colony screening. In particular, the number of clones to screen. So if it's a primary screening or if it's a sub-cloning. So this is depending so on the throughput that you want to achieve and the possibility to have implemented, so to have a compatible and modular automation setup. It's an important aspect to consider. The cell type is also a very important factor, because if the cell line to scale up production could be adherent or growing in suspension, having different nutrient requirement, for instance, or growing for on a coated plates only. These are also aspects to consider. The number of expected clones after the screening is also important, because of course after screening thousands of clones, only really few clones will have the yield of recombinant protein or monoclonal antibody that is desired. The protein localization is also need to be considered, because some proteins can be intracellular localized, or maybe they are cell surface antigens such as GPCR receptor, for instance. And therefore you may need to select an appropriate detection reagent to be able to select positive clones. As we said, another point of the checklist will be if doing a primary screening or a sub-cloning, so which round So if we're doing, again, a primary screening and considering the number of clones that we have to select and to screen for a further downstream approach, such as ELISA or PCR, the screening for highest producer or antigen specificity. So sometimes researchers are more focused on the highest producing clones, or the screening may happen also depending on the antigen specificity, so to avoid any cross-reactivity or unspecific signal. An appropriate fluorescent detection reagent may need to be selected as well, so that to avoid any unspecific signal binding. And finally, also monoclonality verification requirements, which are definitely really important, considering if you want to submit your biology to your biological product to regulatory agencies such as EMA or FDA. And this is an example of how cell type is important, is relevant with the choice of your automated colony picker. So these are pictures representative of colonies of different cell lines. So HEC293 on after 11 days post-plating, or for instance, actually show S cells image a few hours after seeding. So these are important aspects. So you want to have automated colony picker that is flexible, so you can select different type of cell lines growing adherence in suspension or on a feeder layer, such as stem cells for growing on matrigel layer, for instance. The fluorescent detection agent also represents an important selection criteria. So the possibility of having a system, an automated system with different fluorescent filters that allow you with a camera, allowing the detection of fluorescence, such as, for instance, fluorescein or FITC signal, where basically the fluorescent image can be compared to the transmitted light image of your colonies growing over time, and then you can perform your high-value clone selection through the selection, for instance, based on the FITC fluorescence intensity of the clones of interest, which will show up basically as brighter, as the brightest clones, having more halo around the colony, indicating of the presence of the secretion of our antibody, for instance, or recombinant protein. This is also an example. Let's go for the binding specificity screening. So this is also an example on how it's important to select also an appropriate fluorescent reagent, for instance, depending on the type of antigen that may want to be selected. So this is a picture in white light of colonies, showing you here how you can confidently select your antigen when you add the fluorescent detection, basically. So you have on the left a FITC conjugated antigen with fluorescence detection, while on the right the absence of any unspecific signal confirmed by the absence of signal by the control protein. Therefore, this is also an important aspect to consider, so the binding specificity assay when choosing an appropriate detection agent for the automated screening. And therefore, how you will perform the rapid selection of your high producers. So when screening, when performing cell line development, often you will go through the transfection of a gene or a gene fragment into a cell line. into your target cell line, you will see these cells will wait for recovery and seed them to let them grow into colonies, often on semi-solid medium. Therefore, these colonies, which may be CHO cells colonies or hybridoma colonies, may start secreting basically recombinant protein, which can be tagged with a fluorescent detection agent, which can be therefore detected as halo fluorescence, as we have shown before. At the end, so therefore, in the end of this workflow, what is also important will be the monoclonality verification assurance, also happening from day 0. So if you start from single cell isolation on day 0 for dispensing or bioprinting your cell line, you may need to verify the monoclonality, basically, so verifying that you have isolated a single cell, and you want to be able to track the colony outgrowth of this over time. And in order to file these details and picture, you may want to export everything, so all the data that you have selected in a PDF monoclonality report containing data and pictures of your colony to make sure that this colony originated from a single cell. And of course, you may want to be able to do this with white light, but also with a fluorescent imaging, as there are automated imager contributing to with multiple fluorescent channel contributing to the monoclonality workflow and the confluency analysis. So colony picking, so how this colony picking and automation can contribute to the monoclonality verification. With the most recent technologies, as we said, the Z-Stack technology acquisition over multiple focal plane can allow you to reduce the screening time from 2 rounds of cloning to 1 round. So we have a Z-Stack, rapid Z-Stack acquisition, which provides you monoclonality verification with high confidence from day 0. And this system, so the ClonePix 2 with monoclonality verification, represents an all-in-one system to simplify your workflow from the single cell identification to the productivity screening. And this is an example now on how molecular devices can integrate different type of technology within a cell line development workflow. So as we said, if you start with, for instance, molecular cloning, so plasmid DNA production and DNA amplification, you may want to be able to produce and scale up your plasmid production by the use of QPX automated microbial colony picker. Once you have performed this step, basically you want to be able to transfect, for instance, your plasmid into the target cell line and then be able to screen cell clones to identify the highest producers. This could be performed with the ClonePix 2 mammalian colony picker, and therefore colony outgrowth may be and colony formation may be followed up by automated colony imaging with the Clone Select Imager FL, or also for monoclonality verification workflow, the ClonePix 2 technology colony picker with the Z-Stack imaging for monoclonality assurance can be used. However, there are also some other technologies which can allow you to have a more other customized type of clonal screening, such as the ClonePix with customized for stem cell picking. So also this goes also in considering which cell type you may want to use. And also after basically achieving what for instance, a cellular knockout or knock-in, for instance, you may want to be able to make some other cell growth screening or functional screening with a high content imager devices, for instance, for 3D organoids to track also 3D organoids, 3D models as well. Therefore, how we can help our customers to automate an entire cell line development workflow. So as you can see here, these are different instruments that also Janet was presenting earlier, integrating into the same workflow, which can be used also for mammalian clonal screening, in providing the ability to run multiple cell line development projects in parallel and reducing the FTE time by 75%. Of course, in this case, you will have an increased capability to screen hundreds of clones in different months to thousands of clones in 1 month, and it will reduce the error and consumable waste due to manual processes. So another couple of workflows, of course, now we have to so automated colony picking for mammalian cell lines can be also used for hybridoma screening workflow, where of course we have the fusion of myeloma cells with B cells, which now form a hybridoma, which produce a monoclonal antibody. And of course, the throughput required by hybridoma clonal screening is often high, so we need to screen also here thousands of clones in order to be able to identify the best producers. As you may know already, monoclonal antibodies are used as therapeutic for cancer and other different type of applications. So we need to implement, often automation is required to implement this workflow in terms of clonal screening, but also functional characterization with ELISA screening, scale up and winning, and also in terms of expansion of the platform. So another hint towards the future of automated colony picking will be the possibility of reprogramming stem cells, their patient-derived stem cells such as iPSCs, which can be edited and engineered using the CRISPR-Cas9 genome editing technologies, and therefore differentiated in vitro in other cell types. So it's important that automation also considers compatibility with these stem cell types for further investigation within other applications, such as disease modeling and also drug screening. Therefore, that's why at Molecular Devices, we also offer customized solution specific for stem cell differentiation. So will be colony pickers also with customized pins, which are specific to the picking of stem cells, so such as also iPSCs, for instance. And lately, I just would like to give you a hint on why is therefore important to automate workflows. Of course, there's no need to specify that. Of course, the complexities of high throughput workflows create a demand that can be alleviated with automation. So when you're considering to implement your workflow with automation, there are different things to consider. So process improvement. So as we said, the throughput, so in terms of number of plates, number of clones to screens, and so on. But also environmental factors may play an important role. So if the cell line, if the clonal screening or cell line in use is temperature sensitive, gas sensitive, or there are some specific media conditions required. And also another important factor will be the monitoring of production, so the QA/QC, data consistency and data management, and also the workaway time and how to reduce the manual, let's say the manual impact, the manual interaction with this automated platform in order to reduce the human errors. Therefore, good automation, what we can suggest you as a good automation, as we said, would be to increase the throughput, but also offer environmental control. Another important aspect would be improve the space utilization and worker safety, provide a full audit trail, so data traceability will be important, and also the scientific currency in order to achieve high quality data and reproducible data for further publication. And this is how molecular devices can help you providing custom lab automation workflow. So you can integrate, we said, these different instruments, so mammalian and microbial, colony picker, but also microplate reader, imagers, confocal imagers, and which can be basically integrated with liquid handlers and automated incubators, and also robotic arm. And therefore, so these are the biopharma, some of the biopharma instruments, so lab automation with molecular devices, with the set of instruments that are available from molecular devices, so the Clone Select Imager FL and the dispenser, single cell dispenser as a bundle for the monoclonality verification workflow, the QPX series of microbial colony picker for plasmid production or synthetic biology, the ClonePix series mammalian colony picker for clonal screening, high throughput and hybridoma screening, and also the microplate reader SpectrumX ID5 multimode microplate reader, and Image Express confocal HTAI for high content imaging system. And yeah, so I would like to thank you everyone for your attention. Feel free to ask any question. Thank you very much.
Thanks, Carola. There was just a couple of questions in the chat. I'm just going to read those out. The first question was regarding the software. Are there any specific considerations that should be thought about then at that time when you pick the software, or what's important with the software that comes along with a colony picker?
So yes, of course, consideration were going through the software of your colony picker. Yes, of course, you may want to use I would say a user-friendly software which guides you step by step in what you're doing, and also, you know, it should be some user-friendly interface. You should also consider some factors, so which type of imaging detection and imaging settings you want to have, depending of the type of microorganism that you want to investigate as well. So if you're using microbial or, you know, mammalian colony picker, another consideration could be, you know, which magnification you want to have, which type of camera, and also how easy is to interact also with the software from a user perspective, I would say.
And also we mentioned a little bit about the API. So if you're considering to add additional automation, is that something that you could comment on?
Yes, so basically adding additional automation, it means that the system is flexible and modular. So basically you can easily integrate. So if you need to increase your throughput in terms of plates that need to be fed, for instance, in an instrument, and you have a liquid handler performing, for instance, plate dilutions and other type of protocols. And of course, you think about, you may wanna communicate, have communication between the liquid handler and the colony picker, and therefore you can use a robotic arm to to take a plate from the liquid handler and put it directly into the colony picker, for instance. And therefore have some also storage space. So after the colonies are picked, you can put it back into an automated, you can put the plate back into the automated incubator, or maybe also have it in some ambient storage hotel.
Yeah, perfect. And I think I would just add a comment there about the scheduling software of your colony picker provider should be able to handle the communication between the different robotics on your workflow. So that's also something else to consider. And just one last question here about the environmental impact of the labware. Is there any specific way that we can improve the amount of plasticware that we use when we're colony picking?
Oh yes, of course. So minimizing, so we of course right now minimizing the amount of plastic labware is really important because of the footprint, carbon footprint, and for environmental reason. So one of the thing is to reuse some of the reagents, such as within the Cupix colony picker, for instance, the pins can be washed through buffer and are sterilized and can be reused for basically multiple run of colony picking. So that saves a lot of plastic consumables in terms of avoids so skipping the plastic pipette tip step, basically, and ensuring without any cross contamination at the same time.
Great. And how do you ensure that that is sterile? I think I'd just add that comment as well.
Yes, so there are different systems to ensure sterility. So there are wash baths inside the instruments, which can be filled with 70% ethanol, water, and 1% bleach. So different buffers that are regularly used for microbiology sterilization laboratories. There is a halogen lamp, which also ensure a further layer of sterility, and also UV lamp, so for further sterilization, basically.
Great. Thanks, Carol. That brings us nicely up to 11 o'clock. So we've just about run out of time for any more questions, but yeah, any more comments? I'll let go back to Avantor team to round up the webinar. Thank you so much, everyone, for joining.
Thank you very much, Janet. Thank you, Carola. We have still a few questions. I will send them to you, and if we can answer those questions, I will send back to the attendees who asked them.
Yes, of course. Yeah,
So,
no problem.
thank you. It was very interesting, very comprehensive presentation, and thank you all. As I said, the webinar has been recorded. You'll get the link together with some additional documentations after the session. You can also access a replay of this webinar within the Avantor webinars corner. It will be posted here for 1 year, so you can go through it again at your own convenience. I hope this presentation was interesting and helpful, and it has met your